Alright, ji, assalamu alaikum everybody. How are you all? I hope that you all are doing well. Welcome back to my YouTube channel. So today we will be looking at CIE AS level biology paper 9700 paper 2 May June 2025. This paper is of 1 hour and 15 minutes. So let's continue discussing paper 2 of the AS level biology. Let's move on to question number 1 over here. Question number 1 over here says that root hair cells are specialized plant cells located in the outer layer of the young roots of the plants. Root hair cells have an essential role in the uptake of water and dissolved mineral ions from the soil solution. So they are saying basically that the root hair cells are the specialized plant cells that are found in the roots, and they play a role in the uptake of the water and the dissolved mineral ions from the soil solution. Now then further in the question they say that the transport of water across the root to reach the central xylem tissue can occur by the symplast pathway or the apoplast pathway. So we have discussed these two pathways in the transport in the plants chapter. So we are familiar with the symplast pathway and the apoplast pathway. Part A over here says that figure 1.1 is a diagram of a photomicrograph of a root hair cell. On figure 1.1, name the cell structures Z, Y and X. So basically they are asking us to label Z, Y and X. So Y is basically the tonoplast. What is a tonoplast? Tonoplast is basically the cell membrane of the sap vacuole. The vacuole, the large and the permanent vacuole of the plant cell is known as the sap vacuole. And then what is this over here? This must be, the Z must be a nucleus over here, because we know that the nucleus is also found in the root hair cell. Usually location of the nucleus as studied in the books is this, but because it is not here, then obviously it would be here, and it is the nucleus. Now if I generally discuss with you guys the idea of the symplast and the apoplast pathway. So basically what is the symplast pathway and what is the apoplast pathway? In the apoplast pathway, basically it involves the movement of the water through the cell wall, right? It involves the movement of the solution through the cell wall and the intercellular spaces. And if I talk about the symplast pathway, so symplast pathway involves the movement through the interconnected cytoplasm through the plasmodesmata cytoplasm, right? So now let's look at the part B of this question. Part B of the question says that in the symplast pathway, water passes through the cells of the different tissues in the roots before entering the xylem vessel of the xylem tissue. Name the tissues of the root in the correct sequence through which the water passes in the symplast pathway. In the symplast pathway, what happens is that the movement of the water is through the cytoplasm of the cell via the plasmodesmata. Cytoplasm via the plasmodesmata. Question, they are saying that we have to name those structures through which the symplast pathway occurs and the water reaches the xylem. Now I would recommend you all to see this diagram to understand the question further. So they are asking us about the symplast pathway. So let's see the symplast pathway. Basically what happens, what is the symplast pathway over here? That the water enters the epidermis, right? The root hair cell epidermis cells, and then it moves from the root hair cells to the cortex cells, and then from the cortex they move to the endodermis cells, and from the endodermis layer of the cells they move to the pericycle. The water moves to the pericycle layer of cells, and then from the layer of the pericycle they move to the xylem. So this is how the water reaches from the soil. Water from the soil reaches through the whole tissue cell. So this is how the water from the soil reaches via the symplast pathway towards the xylem. So what will be the correct sequence over here? The sequence would be epidermis, right? The sequence will be the epidermis, then the cortex cells, which is also known as the parenchyma cells, then the endodermis, then the pericycle layer of cells, and then the xylem. So this is the whole sequence of the symplast pathway. I hope it's clear to all of you. Now in part C of the question, they are saying that in the apoplast pathway, water passes along the cell walls of the adjacent plant cells and through the intercellular spaces. This is more efficient than the symplast pathway. Explain the structural features of the plant cell wall that makes the apoplast pathway an efficient pathway for the transport of the water. So what happens in the apoplast pathway? Basically the water moves through the cell wall or the intercellular spaces of the cells, right? So for example, over here the water enters the plant cell, right? So it will travel through the cell wall, and then over here the Casparian strip will be there. It will prevent the passage of water further by the apoplast pathway. So the water has to enter the endodermis, and then it will continue its path by the symplast pathway and reach the xylem. So eventually the apoplast pathway has to convert into the symplast pathway uh, to basically carry itself to the xylem. But then in the question, basically they are saying, they explain that what structural features of the plant cell wall makes the apoplast pathway an efficient pathway for the transport of the water. So let's look at this. So the first point we can rise over here and can give in our answer is that the cellulose fibers in the cell wall are hydrophilic. We know that cell wall is made up of the cellulose, right? So cellulose fiber are hydrophilic in nature, which means that they are water loving in nature. Water loving means that the cellulose fibers are going to attract the water molecules. So this attraction will allow the adhesion of the water molecules with the cellulose fibers, and it will allow the smooth movement of water molecules through the cell wall. Another point over here is that the arrangement of the cellulose fibers leave the spaces and pores between them. For example, this is the structure of the cell wall which I have taken from the internet, and if you guys can see over here, these are the cellulose microfibrils, and these are leaving the structure of the cellulose microfibrils is such that it leaves the spaces and pores between them. For example, if this is a cell wall and this is the arrangement of the cellulose fiber, so it is leaving some pores between them. So these pores will act as the channel for the free water flow between the cells, and we can write it over here as the arrangement of the cellulose fibers leave the spaces for the water to move through, right? So this offers a path of least resistance for them to move. And then another thing over here is this, that another point you can say over here is this, that the cell walls, unlike the cell surface membranes, are freely permeable. Cell surface membrane, if you guys remember, the cell surface membrane are the selectively permeable membrane, right? They are selectively permeable membranes, right? But this is not the case for the cell walls. Cell walls are freely permeable. So as the cell walls are freely permeable, so it provides a path of the least resistance, allowing a rapid and the continuous water flow. So this will also offer a path of the least resistance, and it will allow the water to flow freely between the cells as well. So this is basically how you can write a structured answer for this part. Now let's move on to part D over here. Let's give part D a read over here. It is a lengthy part. Root hair cells also synthesize and secrete substances into the soil. Electron microscopy of the structure of the root hairs have identified the endoplasmic reticulum and a number of small Golgi bodies and numerous vesicles in it. Root hair cells of the barley plant secrete enzymes known as the acid phosphatase, which catalyzes the release of inorganic phosphate ions from the organic phosphates in the soil. So basically they are saying that in the root hair cells, let's summarize the question as well, and then let's summarize that what they are asking. Let's summarize the information that they are actually providing us in the question. They are saying that in the root hair cells they have seen the endoplasmic reticulum. Now the endoplasmic reticulum can be the rough endoplasmic reticulum and it can be the smooth endoplasmic reticulum. Then they are talking us about the two type of the plants, right? Root hair cells of two types of the plant. One is the barley plant, right? And they are saying that it secretes the enzyme known as the acid phosphatase. Then next they are talking about the root hair cells of the sorghum plant, which secretes the hydrophobic lipid compound which is known as the sorgoleone, which slows down the growth of the neighboring plants. So basically the another plant we are having is the sorghum plant, which secretes the lipid compound. Now in part 1 they are asking us that explain why the proportion of the rough endoplasmic reticulum and the smooth endoplasmic reticulum may be different in the root hair cells of barley plants compared with the root hair cells of the sorghum plants. Now in order to basically answer this question, we must know the function of the rough endoplasmic reticulum and we must know the function of the smooth endoplasmic reticulum. Now if I talk about the rough endoplasmic reticulum, so the one-liner function for the rough endoplasmic reticulum is the protein synthesis, right? And for the smooth endoplasmic reticulum is the lipid synthesis. Now in the question they are saying that why the proportion of the rough endoplasmic reticulum is higher in the cells of the barley compared with the root hair cells of the sorghum plant. So basically in the barley plant what happens is that they are secreting the enzymes, right, which is known as the acid phosphatase, and we know that the enzymes are the proteins. We know that the enzymes are proteins and proteins are synthesized in the rough endoplasmic reticulum. So that's why, because they are secreting the enzymes which are known as the acid phosphatase, and enzymes are protein in nature, so that's why the rough endoplasmic reticulum is more in the barley plants. And then what happens in the sorghum plant, that they are secreting the lipid compound, and lipid compounds are synthesized by the smooth endoplasmic reticulum. So in the sorghum plant the SCR would be more. So we can write our answer in a way that why the rough endoplasmic reticulum in the barley plants are more as compared to the sorghum plants. It is because they secrete the enzymes, right? And enzymes are protein in nature, and rough endoplasmic reticulum is the site of the protein synthesis. We can write it in this way. Also, if we want to go vice versa, we can say that the sorghum root hair cells have more SCR because they secrete sorgoleone, that is lipid in nature. So that's why the SCR is more over there. D part 2. D part 2 over here says that student X stated that acid phosphatase, which is the enzyme, and sorgoleone could be transported out of the root hair cells using the same processes. Sorgoleone was basically the lipid compound which was released by the sorghum plant, which was secreted by the sorghum plant basically. Now the student Y stated that the acid phosphatase and sorgoleone are transported out of the root hair cells using different processes. Now student X is stating that it can be transported out by the same process, and student Y is stating that it can be transported out of the root hair cells using the different processes. And then they say that suggest the reasons given by student X and student Y to support their statement. So basically if they are asking about the same processes, so acid phosphatase is the enzyme which is protein in nature, and this is lipid in nature, and if these both are to be transported out of the root hair cells by the same process, so it must be the exocytosis, right? And if it must be transported using the different processes, so it can be simple diffusion as well, right, or by the transport proteins as well. Like for example, if sorgoleone is a lipid compound, so usually non-polar compounds, small and non-polar compounds are crossed by the cell membrane without any transport protein, and acid phosphatase can use the simple diffusion, right, and acid phosphatase can use the transport protein. So let's discuss it कि किस तरह से we can write the answer. Student X is saying that the acid phosphatase and sorgoleone could be transported out of the root hair cells using the same process, and how it can be done, that both acid phosphatase and sorgoleone could be secreted out by the exocytosis, which is a type of the bulk transport, right? So this allows the bulk transport or the release of large quantities out of the cell. So molecules may be too large to leave by the diffusion, because this is a suggestive question, right? Because this is a suggestive question, so we have to suggest the answer. So this is a suggestion we are giving. Molecules, so we are suggesting that maybe, and we are assuming that maybe the molecules may be too large to diffuse out by too large to leave by diffusion. So they are packed into the vesicles via the Golgi vesicles, obviously, and then they are fused with the plasma membrane and release their content out of the cell, right? And then the vesicle will go fuse with the plasma membrane and release their contents out of the cell. And then what is the student Y basically predicting that the acid phosphatase, as it is an enzyme, it is a protein, so it is hydrophilic in nature. We know that enzymes are hydrophilic in nature, so they cannot cross the hydrophobic bilayer of the cell surface membrane directly. So it requires a transport protein to be secreted out of the cell. Similarly, sorgoleone, which is a lipid compound, and lipids are hydrophobic in nature, so what will happen that as they are non-polar, so they can diffuse through the phospholipid bilayer without the transport proteins or the vesicle. So they can simply diffuse out of the phospholipid bilayer without any transport protein or any vesicle. So these are the two suggestions which student X and student Y are giving according to the transport of the acid phosphatase and the sorgoleone. Now let's move on to question 2 over here. Question 2 over here says that the transport of the respiratory gases involves blood plasma and the red blood cells. Red blood cells contain the globular protein haemoglobin. Now part A over here says that describe the features of a hemoglobin molecule that are typical of a globular protein. So basically they are saying that we have to describe the features of the hemoglobin molecule that are also of a typical globular protein other than having an approximately spherical shape, right? So as hemoglobin is a globular protein, so there is one side which is facing the aqueous environment of the cell, and then there is one side which is facing the inner environment of the cell. So the distribution of the amino acid on the hemoglobin globular protein must be in a way that the hydrophilic amino acids, which are the amino acids that are water loving, right? So they are facing towards the aqueous environment, and the hydrophobic amino acids are tucked inside, right? And this helps in stabilizing the protein core. And also because of this arrangement of the hydrophilic and hydrophobic amino acids in this way, it causes the hemoglobin to become soluble in water as well, which is essential for it to circulate in the blood plasma. And as the hemoglobin molecule will circulate in the blood plasma, then only it can deliver and transport the oxygen throughout the body. Also, unlike all the other fibrous proteins, one is the globular protein, right, and the other one is the fibrous protein. Unlike the fibrous protein, जो globular proteins होते हैं, they have the metabolic roles in the body as well. For example, जो hemoglobin है, it is a globular protein, so it has a role in the transport of oxygen and the carbon dioxide, right? So yes, this is the explanation for this part. And now how we have to basically write the answer for this part, it is as that the hemoglobin has the hydrophilic amino acids on the outside, which is facing the aqueous environment, and the hydrophobic amino acid on the inside, making it soluble in water, making it stable and able to function in the oxygen transport. It means that because of this structure of the hemoglobin molecule as a globular protein, it can transport the oxygen well. Now let's look at part B over here. Part B over here says that a number of substances are involved in the transport of the respiratory gases. Complete table 2.1 by stating the name of the substances that matches the description of its role in the transport of the respiratory gases. The first row has been completed for you. So this part of the question is from the transport in mammals chapter. Transport in mammals chapter, right? So it is about the respiratory gases and the substances involved in it. Now the role in the transport of the respiratory gases, we have to see this column and then we have to answer the substances over here. In the capillaries of the respiring tissue, this combines with the water to form the carbonic acid. So we know that in the respiring tissues, the carbon dioxide is uptaked, right, and transported in a form of the carbonic acid, right? So carbon dioxide basically combines with the water to form the carbonic acid H2 CO3, right, in the presence of the carbonic anhydrase. So this is that in the capillaries of the respiring tissue, this combines with the water to form the carbonic acid. So it would be the carbon dioxide. So that's why they have written carbon dioxide. Now we have to do the similar. We have to solve the similar rows according to this. The next statement over here is saying that in the capillaries of the respiring tissue, this enters the red blood cell through a membrane transport protein. Now basically what happens in the capillaries of the respiring tissue, that in the respiring tissues the carbon dioxide is produced as a waste product of the cellular respiration, right? And then what happens is that this carbon dioxide basically diffuses into the capillaries, right? It diffuses into the capillaries and to be transported to the lungs for the excretion, right? So the carbon dioxide should not stay in the blood for a longer time, as it is already very toxic to the blood, right? So what will happen, that the carbon dioxide will enter the red blood cell and it will combine with the water to form the carbonic acid, right? And then the carbonic acid basically it will form the bicarbonate ions, right? What it will do, that the carbonic acid will form the bicarbonate ions and the H positive ions, bicarbonate ions and the H positive ions. Now the problem over here is this, that if too much of the bicarbonate ions will be built up inside the red blood cells, then the charge balance would be disturbed, because it is an active respiring tissue, so it would be producing a lot of carbon dioxide, right? And then a lot of carbon dioxide would be entering the red blood cell and then forming the bicarbonate ions. And if too much of the bicarbonate ions would build up in the red blood cell, then the charge balance is disturbed. So for example, if this is a red blood cell and this is a transport protein over here, so to maintain the electrical neutrality, what will happen, that the bicarbonate ion is going to diffuse out of the red blood cell and the chloride ion is going to enter the red blood cell through the same transport protein, right? So in the capillaries of the respiring tissue, this enters the red blood cell through the membrane transport protein, so this will be the chloride ions. In the red blood cell, this is formed when carbon dioxide binds to the hemoglobin. Now we over here know that there are three methods through which the carbon dioxide is basically transported from the tissues to the lungs. So the 5% of the carbon dioxide dissolves in the blood plasma while the remaining diffuses into the red blood cell. So the 5% is dissolved in the blood plasma. Then the 10% of the carbon dioxide, we know that it binds to the amine group at the end terminal of the polypeptide chains of the hemoglobin in the red blood cell. So when the carbon dioxide basically combines with the amine group at the end terminal of the polypeptide chains of the hemoglobin, so it forms the carb amino hemoglobin. So when carbon dioxide directly bounds to the end terminal amine group at the end terminal of the polypeptide chain of the hemoglobin, so it forms the carb amino hemoglobin. So the answer over here would be carb amino hemoglobin when carbon dioxide binds to the hemoglobin. Whenever the carbon dioxide would bind to the hemoglobin, it would bind at the amine group of the polypeptide chain of the hemoglobin. So the answer over here is the carb amino hemoglobin. Now the next pointer over here is this, that in the alveolar capillaries, this combine with the hydrogen ions in the red blood cell to form the carbonic acid. So what will happen, that we know that H positive ions are going to combine with the HCO3 minus and it is going to form the H2CO3, which is the carbonic acid. And then in the alveolar capillaries, what will happen, that this carbonic acid is basically dissociated into the carbon dioxide plus water, and then this carbon dioxide is diffused out of the alveolus or the alveoli, and then it is excreted out through the nose. So this is how the carbon dioxide is excreted out. So the answer over here would be the hydrogen carbonate ions. Yes, hydrogen carbonate ions. because these are the ions which are combining with the hydrogen ions in the red blood cells to form the carbonic acid, and then this carbonic acid is dissociated into carbon dioxide and water, uh, and then the carbon dioxide is diffused out of the alveoli. So the answer over here would be the hydrogen carbonate ions, right? And let's come to the next row. The next row over here is saying that in the alveolar capillaries, this is formed in the red blood cell as a result of oxygen binding to the heme group of the hemoglobin. So we know that there are 4 polypeptide chains in the hemoglobin, right? And there is a central heme group which contains the iron Fe plus 2. These are the 4 heme groups which contains a central iron Fe plus 2. So what will happen that the hemoglobin is basically going to combine with the oxygen to form the oxyhemoglobin, right? Oxyhemoglobin. So this is formed in the red blood cell as a result of oxygen binding to the heme group. So it would be the oxyhemoglobin. The answer would be oxyhemoglobin, right? And then the next over here says that in the red blood cells, this combines with hemoglobin to form hemoglobinic acid. So we know what happens is that the carbon dioxide basically combines with the water to form the carbonic acid in the presence of the carbonic anhydrase enzyme, right? So what is going to happen that because it is an acid, so it would be acidic for the cell. So what will the body do that it will dissociate this into the H plus and HCO3 minus, which is the bicarbonate ions and the hydrogen ions. Now the bicarbonate ions are going to move, if they build up in the cell, so it is going to move out in the blood from the red blood cells, right? And the chloride ions are going to move in while these are going to move out, right? As these will move out, so the chloride ions will move into the red blood cell to maintain the electrical neutrality. Now what will this H positive do? H positive is also acidic, right? So hemoglobin, we all know that hemoglobin acts as a buffer. So what is going to happen that the H positive ions is going to combine with the hemoglobin to form the hemoglobinic acid. So this will be the hemoglobinic acid. So in the red blood cell, this combines with the hemoglobin to form the hemoglobinic acid. So hydrogen ions basically combine with the hemoglobin to form the hemoglobinic acid. So the answer over here would be the hydrogen ions. I hope that this part is clear to all of you. Now let's move on to question number 3 over here. Question number 3 over here says that the carrots are the root vegetables of the carrot plant. The carrot plant is an important food crop that is grown throughout the world. Carrot have a sweet taste because sugars form a proportion of the total carbohydrate present. Plant breeding has produced many different varieties of carrot with different levels of sweetness. Alright, so they have just told the story in the initial part of the question. Now let's come to the main part, which is A part. One of the sugar found in the carrots is the galactose. Galactose has the molecular formula of this C6H12O6 as the alpha glucose. This is the general formula for the carbohydrate glucose. Figure 3.1 shows the molecular structure of the galactose found in the carrots. This is similar but not identical to the molecular structure of the alpha glucose. Galactose is also a monosaccharide. It is also a monosaccharide. In figure 3.1, the 6 carbon atoms are numbered 1, 2, 3, 4, 5, 6. Over here, if you guys can see 1, 2, 3, 4, 5, and this is 6. Now, what they are saying over here is this that now what they are basically asking us to do in the question is that state the differences between the molecular structure of the galactose shown in figure 3.1 and the molecular structure of the alpha glucose. Now in your syllabus you must know the structure of the alpha glucose and the structure of the beta glucose, and you must have studied it in the chapter of the biological molecules. You must remember its structure. It is not a very difficult structure. So you guys must remember the structure of the alpha glucose and the beta glucose. Now they have given you the structure of the galactose and they have asked us that tell us the difference between the alpha glucose and the galactose. So if you see over here, so in the C1, on the position of the C1, you can see that the hydrogen and the hydroxyl group are basically the other way around. In it, the hydroxyl group is up the chain, and over here the hydroxyl group is below the chain. And in the C4 also, you guys can see that the hydroxyl group and the hydrogen group are the other way around, right? Over here the hydroxyl group is up and H is below, and over here also over here the hydrogen group is up and the hydroxyl group is below. So how we can write its answer? We can write its answer in a way that OH and H group are other way round in at the position, not in, at the position C1 and C4. So, and also this is a question for 1 marks, so this answer is enough to secure full marks. Now let's move on to part B over here. Part B over here says that an investigation was carried out to determine the sugar content of mature carrots produced by different local varieties of the carrot plants that are grown in Tunisia, North Africa. Carrot plants were grown from the seed under the standardized condition. When mature, the carrots were harvested. The different sugars present in the carrots were identified. Measurements of the sugar content of each of the sugar were made. Table 3.1 shows the result of 5 different local varieties of the carrot plant A to E. So basically they have gathered a local variety of the carrots A to E, and they have identified the sugar content of the carrots, right? After studying the results shown in table 3.1, a student concluded that the carrots from the local varieties contain the same 4 sugars, right? The student made 3 conclusions from the data in table 3.1. Conclusion 1 over here is that there are non-reducing and reducing sugars in the carrot. So the conclusion 1 is that there is the non-reducing and reducing sugars in the carrot. So fructose, galactose and glucose, they are the reducing sugar, and we all know that sucrose is a very popular non-reducing sugar, right? And then the conclusion 2 over here says that there are monosaccharides and disaccharides. There are monosaccharide and disaccharide sugars in the carrot. So monosaccharide sugar, glucose, fructose and the galactose, they all are the monosaccharide sugar, and sucrose over here is the disaccharide sugar. Sucrose is the disaccharide sugar. And if I talk about that, the what monomers is the sucrose basically made up of? So sucrose is made up of the fructose plus the glucose. So sucrose is a disaccharide sugar over here. So this conclusion is also being supported. Then if I move on to conclusion 3 over here, the carrots have the same pattern of results. So for it we have to study the pattern. Now let's look at the further question. They are saying that explain the evidence in the table 3.1 that supports the 3 other conclusions. So if the conclusion 1 was that there are non-reducing sugars and the reducing sugars in the carrot, so we can say that yes, the all varieties contain the fructose, glucose and the galactose, which are the reducing sugars, and sucrose, which is a non-reducing sugar. So all of the varieties are containing the glucose, fructose, galactose and the sucrose also, which is a non-reducing sugar. Then the conclusion 2 over here says that there are monosaccharides and disaccharides. So yes, the all varieties contain the fructose, galactose and glucose, that are the monosaccharides, and sucrose, which is a disaccharide. So this conclusion is also supported. Then in the conclusion 3, they are saying that the carrots have the same patterns of the result. If I talk about the highest percentage of the sugar, if I talk about that which is the highest sugar present in the carrot, so I believe it would be fructose, right? Fructose is highest in A, B, C, D and E. Fructose, the amount of the fructose is highest in all of the sugar, and the lowest sugar is the galactose in all of these 5 varieties. So we can the conclusion 3 is basically supported, that all the varieties show the same order of sugar content, with the fructose being the highest and the galactose being the lowest. So the galactose is the lowest sugar in all the 5 varieties, and the fructose is the highest sugar in all the 5 varieties. So it is concluded that yes, they follow the same pattern of results. So I hope that this part is clear to all of you. Now in the C part, they are saying that the carbohydrates that are not sugars are also present in the carrot. Name one carbohydrate that is present in the carrot and that is not a sugar. So it can be cellulose over here, because carrots contain the plant cells, and plant cell wall is having cellulose. Now let's move on to the part D over here. Part D over here says that each carrot plant produces a carrot in the first year of the growth. If the carrot is not removed from the plant after it matures, the plant passes through a dormant period and shoots develop from the carrot in the second year of the growth. This allows the flower to be produced and the seed formation to occur before the plant dies. Explain with reference to the life cycle of the carrot plant when the carrot acts as a source and when the carrot acts as a sink. If I talk about the general definition of what is a source, so basically source refers to that part of the plant that produces the sugar, right? That produces the sugar. And the sink refers to that part of the plant that utilizes that sugar or stores that sugar. Utilizes or stores the sugar, right? So now they are saying that with reference to the life cycle of the carrot plant, we have to explain that when the carrot will act as a source and when the carrot will act as a sink. So let's talk about that. When does the carrot acts as a source? So source is the carrot acts as a source after the dormant period, means that in the second year when the shoots develop. So if the carrot is left in the ground, the plant becomes active in the year 2. So what will happen, that the stored reserves in the plant will become active in year 2, and the stored reserves in the carrot are mobilized. What does it means by the mobilized? It means that the starch is basically converted into the starch which was stored in the carrot is converted into the soluble sugars, and then these soluble sugars are then loaded into the phloem and transported to the developing shoots, flowers and seeds. So at this stage the carrot acts as a source, providing the assimilates for the growth. So we can write this in a form of an answer in a way that because the stored reserves, so we can write the answer of this in a way that because the stored reserves are mobilized, like starch is basically converted into the soluble sugar, and these soluble sugars and that are then loaded into the phloem, and then from the phloem they are transported and then from the phloem they are transported to the developing parts of the plant, like for example the flowers, the seeds, the shoots. And then if I talk about the sink over here, so in this and then if I talk about the sink over here, so sink is in the first year, right? When the carrot is forming and enlarging, so the carrot is acting as a sink. How the carrot is acting as a sink? Because during the first year the carrot root is a growing and a storage organ that receives the assimilates, for example the sugars and the amino acids that are produced by the leaves. So these assimilates are transported to the phloem and then they are unloaded in the roots, where they are converted to the storage compounds, for example the starch, right? So the carrot acts as a sink. So we can write the answer in a way that because it receives the assimilates sugar via the phloem for the growth and the storage, so that's why the carrot is acting as a sink. Now let's have a look at part E over here. Part E over here says that carrot virus Y is the pathogen of the carrot plants. The virus, which belongs to a group known as the Potyvirus, replicates its viral nucleic acid and proteins within the host carrot cells. The general structure of the viruses is shown in figure 3.2. So this is the carrot virus Y, which is the pathogen of the carrot plant. So this is the general structure of that virus. Then further in the question they are saying that the synthesis of the viral proteins in the host carrot cells only involves the process of translation. Translation means the protein synthesis, right? Translation means the protein synthesis. The process of transcription does not occur suggests why the translation occurs in the host carrot cells during the synthesis of the viral proteins, but transcription does not occur. So first of all, you must understand that what basically transcription is. So transcription is basically when a specific part of the DNA is transcribed into the mRNA, right? So this is known as the transcription. Now in the question they are asking that suggests why the translation only occurs in the host cell and why the transcription is not occurring. So we can give our answer in a way that this structure of the virus is having the RNA. It is not having the DNA. So since it is not having the DNA as the genetic material, so they do not need the transcription, right? What happens in the transcription, that they normally in the transcription they make mRNA from the DNA, right? And as the DNA is not present in this structure of the virus, so there is no need for the transcription. Their RNA genome is already acting basically as the mRNA, and it carries the codon sequence that is basically needed for the direct protein synthesis. So therefore you can say that the viral RNA can bind directly to the host ribosome to be translated into the polypeptide without requiring the transcription first, right? So how we can answer this question? So we can answer the question in a way that the virus has the RNA as its genetic material, so no transcription is needed. The viral RNA itself will act as the mRNA. It will bind to the host ribosomes, and we know that when the mRNA binds to the ribosome, then the protein synthesis takes place. So when the viral RNA will act as the mRNA and it will bind to the host ribosomes, so mRNA, and it will bind to the host ribosomes, and then it will directly be translated into the proteins. So this is how you are going to write down its answer. So this was all about May-June 2025 paper 2 yearly past paper part 1, where we covered question 1 to question 3. Now in the next part we will cover question 4 to question 6. If you guys benefited from the video, then do remember me in your prayers. Allah Hafiz everyone, take care, see you guys in the next part.